Great. Thank you for welcoming me to your lab meeting. I appreciate it. Oh, wait, what am I doing? I have to share my screen first. All right, so you should see my slides now. Oh,
Perfect.
great. So yeah, thank you very much for welcoming me. And I know there's some potentially some common interest in these kind of themes. I'm just going to talk about one particular project, as the title says. We're looking at the evolution of neural activity leading up to a spontaneous voluntary movement. I have here a case study in perseverance. I mentioned that this is work that I started with Rob Shapire, who is one of the co-developers of AdaBoost, at Princeton, and his then PhD student Mehmet Basbug. Mehmet has since gone on to he's working for a startup, and my PhD student, my current PhD student Luca, has taken over leading the project. But it is something that we literally started. I think the conception of this goes all the way back to 2012. We've been working on it for a while. But I'm happy to say we've finally, I think, rounded the bend, and we're getting ready to actually submit this, or resubmit it. So it was in review at PNES, and the reviewers asked for more data, and COVID came, and then we had other problems. We found bugs in the software, and et cetera. But I think we've rounded the bend. So the theme here, when I say impending movement, I'm really talking about a spontaneous voluntary movement, and that's the phrase that I tend to use. Here's an example, like you have the diver on the diving board, and you can say, well, yes, there are constraints here. The diver's not totally free, and he's on the diving board. He's clearly expected to dive, so on. There's this whole context that constrains things, but at some point, there's a time scale at which there's a decision to be made, like jump now, and that's not as constrained. So there is some room for what you might call a free decision here, that's not constrained in any obvious way by anything going on locally. So you have that brings up questions like, well, why was this movement initiated exactly when it was, and not say 500 milliseconds earlier or later? How do those kinds of decisions get resolved in general when there aren't any obvious constraints or imperative to move? So just to be clear about what we're interested in, it's something that I refer to as the neural decision to move. And if such a thing does exist, and I'm not necessarily saying that it does, but if it does exist, what we're looking for is this proximal neural event that commits some part of the body to an imminent movement. That's sort of what we're after. And that question is pertinent in a lot of different contexts. I throw up a few of them here, not least of which is developing brain-computer interfaces. So they have, they've made incredible strides, especially now that people have started to look at invasive methods. But there's one of the most challenging, outstanding problems is not to know what the user intends, but to know when, to be able to detect in continuous time, like when the user wants to do something. That's more of a challenge. And if you think about it, well, think about a brain-computer interface rather than one that's controlling a robotic arm, like in the picture here. Imagine one that's, you're controlling your word processor on your computer, Microsoft Word. So if you're an able-bodied person, you might be sitting at your computer with your hands like rested on your keyboard, and you're thinking about what you want to write. And if you're like me, you might sit there for a very long time, stuck, just thinking about what you want to write. But you're thinking about what you want to write, but then at some moment you start typing. And so there's the actual fact of, okay, now I'm doing this. And with the brain-computer interface, which is driven, as we say, by thought, how is that interface to know when you're just thinking about what you want to type versus really meaning to actually type now? And that's this streaming problem, or this problem of detection in continuous time, knowing when a person really intends to do something. It isn't just thinking about it. So you may or may not be familiar with this research, but you've probably heard of the readiness potential. So research on this general theme arguably started back in 1965 when Kornhuber and Deka published this account, published their discovery of this cortical electrical potential called the readiness potential, or the Bereitschafts potential, that you get when you record using EEG, in their case, from these these central sites, roughly over the motor cortex, and you see this very slow buildup of scalp electrical potential beginning like 1 second or even more before the onset of a spontaneous movement. So the people were just instructed to kind of make a spontaneous movement every so often, right? They're not too precise instructions. And I think they were told to like try to wait at least 10 or 15 seconds, but not too long, not too much more than that, but to avoid counting time, just kind of wait and just at some point spontaneously move. The reel-to-reel tape here in the background, by the way, that's just a nod to like the low-tech methods they had at the time. So they had a reel-to-reel tape device that was recording the EEG, and then when the person moved, they would stop it, and then they would flip the tapes over and play it back in reverse to capture what had happened in the few seconds before the movement. So Kornhuber and Deka dubbed this the electrophysiological sign of planning, preparation, and initiation of volitional acts. It's the readiness potential, and it's that interpretation has held more or less ever since. And this, what I call pre-movement buildup, it really doesn't matter how you measure it. You can certainly measure it. It's very reliable, measured with EEG, but it's also been measured at the level of single neurons, even in humans. So these were epilepsy patients, and they were able to do it. Itzhak Fried and colleagues did a similar task with human patients, and recorded in many different areas. Here's in the pre-SMA, a slow buildup preceding the onset of movement. So it doesn't matter how you measure it, you get this slow buildup, and it's not just in humans. So you get this pre-movement buildup in primates, rodents, and even invertebrates, even crayfish. And there's some, I think, I've heard some recent work in progress looking at similar slow buildup in fruit flies, I think. So about what was it, 30 years after Kornhuber and Deka, there was this famous study by Benjamin Libet, which probably most of you have heard of. So he did the same thing. He recorded the readiness potential while subjects were making spontaneous voluntary movements, but then they had to estimate when they felt the urge to move each time they made a movement using this fast rotating clock. And the upshot of that was that the conscious decision, at least as timed using this clock method that Libet devised, averaged out. I mean, the variance is quite broad, but it averaged at about 200 milliseconds before movement, whereas you could see this readiness potential stretching back up to a second or even more before movement. And they often talked about the onset of this thing, even though it really doesn't look very linear. It looks pretty nonlinear. So if you kind of follow it back in time, it's not clear that there's any sort of an onset, but you can say, well, the first point at which it was, detectable above the the noise for, I guess. So these two studies, so these two findings, the Kornhuber and Deka and then Libet's, led to what I caricature here as the prevailing view, which is that somewhere far back in time on neural terms, you know, 1 whole second or even more than that, there's this decision. There's this neural decision to initiate movement. And then the rest is the outcome of that decision. So this readiness potential that is like coursing towards an eventual triggering of movement. And then as an aside, right, there's this later event, which is when the self becomes conscious of the decision to move, which you can really take with a grain of salt. It's like unclear what that really means, considering that it's a post hoc judgment that the subject makes like a second or two after they move and so on. It's difficult to interpret, but this is, I would say, a caricature of the prevailing view. What I've worked on leading up to the work I'm describing here, or that I will be describing here, is a different account of the readiness potential that is based on there being spontaneous ongoing fluctuations in neural activity in the motor system, probably in all systems, and that ongoing activity is arguably just noisy. It's stochastic, at least from our relatively uninformed point of view. Maybe if we knew more, we could call it something else, but it seems stochastic. And noise in neural populations and in even at the single neuron level and at higher levels, like in the residuals around reaction times or other behavioral phenomena, even if they are noisy, the kind of noise it's not like white noise, so it's autocorrelated. It's 1/f noise, and that kind of noise is primarily driven by sort of lower frequency fluctuations, and there's ever less energy in the higher and higher frequencies, right? And so the idea was simple. It was just, well, what if, you know, in a context where you don't have a very strong imperative to move at a particular time, then in that context, maybe this ongoing ebb and flow of activity in the motor system actually starts to play a bit of a role in, at the very least, determining the precise moment at which a movement is initiated or triggered. So if there is a threshold at which in the system that leads to triggering the movement, which I have illustrated here as this dashed red line, you know, you might have an inclination to move sometime soon, but the precise moment isn't really fixed. Then the noise might help to determine the precise moment at which the threshold is crossed. And then if I time lock to that threshold, it's kind of a no-brainer. I'm going to see the profile, you might say, of that 1/f noise. In fact, it turns out if you look at this carefully, it actually gives you an estimate of the autocorrelation function if you time lock to these crossings and average over many of them. So this led to what I call the stochastic decision model. The premise is just that when the imperative to produce a movement is weak, then the precise moment at which the decision threshold is crossed is largely determined by spontaneous subthreshold fluctuations. Neural activity and those fluctuations get caught in the event-locked average, looking like a gradual buildup. And I modeled this using a simple leaky stochastic accumulator, which is just basically a drift diffusion model, but with this leak term. And that the leak actually makes it more sort of biologically plausible. And in this case, in the case of my own work, it ended up being necessary to adequately model this or to fit the data. So this is work that we published back in 2012 that sort of upended the going interpretation of the readiness potential, and hence the interpretation of Libet studies and so on. We could argue then, here we could offer that with this new perspective that this slow buildup that we see maybe is accounted for by spontaneous fluctuations, and the actual neural decision or the commitment to initiate a movement doesn't really happen until quite late in the game. And we've called this a late decision account, contrast it with Libet's account, which is an early decision account, saying that the decision happens at the onset of the readiness potential. That's an early decision account. And here we're saying that the actual decision or the commitment to move happens late. So we distinguish between late and early decision accounts. So one of the reasons that this even came up and is even a problem is because of the way that people do research on spontaneous voluntary movement. You need epochs. You need to average because the data are quite noisy. Even single unit data are pretty noisy. So you need to average. And so what do? You have to find some events, and then you time lock to those events. You extract epochs, and then you average them together. And of course, the events that you time lock to, here are these blue arrows, are the movements. That's when you initiate a movement. So you have this sample of data epochs that basically all of them terminate in a movement. Well, that's a very biased sample. It's inherently biased because of the nature of the question and the nature of the way you do the data analysis. But if you think about it, it's a bit like if you're asking like, well, what's predictive of movement, which is kind of essentially the question. It's like trying to learn to understand and predict the onset of rainfall by looking at just a sample of rainy days. So that'd be a very biased sample. You don't know what happens and the degree to which whatever you see in the data is happening even when it doesn't rain. So the caveat is that it's sampling the conditions that are active when a movement is not about to happen is integral to being able to reliably predict and understand what's going on when movement is going to happen. But this is the way that this kind of research has gone on since its inception. We always just look at epochs that are time locked to the movement because that's the event, the only event we have in the data to time lock to. Ideally, you'd want this, right? You'd want some epochs without movement, epochs with movement, compare, see what's going on, but we never have that. One thing that people have done to get around this is, you have just these epochs with movement, but what you do is you take some time window that's quite early and you call that the baseline, and then you can maybe look at other time windows closer and closer to movement and compare those, right? But that's also very biased because the baseline has a fixed and reliable temporal relationship with the onset of movement. So that's problematic as well. We'll see. I'll show you that, some of what we've done. But so this work really started with the desire to do this, to have these somehow have epochs without movement, have a proper control condition, with the prediction that if this late decision account is correct, then distinguishing between these should be challenging, if not impossible, well before the decision, the putative decision. So again, this is work I initially started with Rob Shapiri and Mehmet, and then Luca came on board more recently. We tried to map the time course of neural activity leading up to a spontaneous voluntary movement using boosting, using AdaBoost in this case. I collected some MEG data, simultaneous MEG EEG data from subjects. This is when I was working at Neurospin, just near Paris, France. This is with Stan DeHaan's group. And I had, one of the features of AdaBoost is that it works quite well and provably so, assuming sufficient amount of data. So I had a small number of subjects, but I had them come back for multiple sessions. So I had a large number of data epochs for each of a few subjects. We later added more subjects with fewer sessions, but that's how we started. And the paradigm that we developed was quite simple. What you do is, as a subject, you're looking at these photographs of nature scenes, outdoor scenes or plants and stuff. Nothing that has any, any strong valence to it or anything. Just pretty pictures to keep you engaged. Except there are two kinds of viewing, two kinds of, let's call them trials, because what you're really doing in this task, it's just a slideshow for you to enjoy. That's it. It's just that you may or may not be in charge of when the slide advances to the next one. So before each slide, you get a keyword, either manual or automatic. If you see the cue manual, then that means, and there's a fixation point on the image. So because with EEG and MEG, eye movements introduce big artifacts. So we have a fixation point. On a manual trial, you're instructed to just look at the image and enjoy it for a few seconds, and then just when you're ready, you make a movement. You normally just press a button, and then that advances to the next slide. On these automatic trials, the instruction is just to relax, and the slide will advance by itself after a few seconds or after a little while. And crucially, the viewing time on these automatic trials is drawn from the subject's own viewing time distribution on their manual trials, so that by the end of the experiment, these two kinds of epochs are relatively well matched for how long you were waiting for the slide transition. So this gives us our matched control condition. We have one set of data where we align the data now to the slide transition. And in one case, that slide transition is accompanied by a movement, because the movement was what triggered the slide transition. And then in this other set of data, there is no movement. So you have movement, no movement. And then what we do is just build a classifier using AdaBoost that in a sliding window is trying to tell these two apart at each point at each position of the sliding window over the time period of an epoch that goes back about 3 seconds, right? And we align everything to the leading edge of the window, because we want to know, right, what the classifier had access to before, right, this point in time. And we were very careful not only to align the leading edge, but any kind of filtering that we did or anything involving wavelets or anything like that, we made sure that it was, like, if we used a high or low pass filter, we made sure it was causal, so that no information ahead in the future with respect to the leading edge of the window could leak back in to the window. So really, the classifier is telling you, this is up until now, this is what I can tell you. This is how I can predict between manual and automatic. For those of you who don't or aren't familiar with AdaBoost, the way AdaBoost works in a nutshell is to take usually a quite large number of very weak, simple classifiers, and learns how to combine their output, or let's call it their opinions, in just the right way, so as to build a very powerful classifier from the individual opinions of a large number of very weak classifiers. That's the essence of boosting. What we used as features are these Haar wavelets, look like this, and there's one that's missing here, which is just a flat line, which is just basically an offset, and that one does actually become important. So we have basically 3 kinds. We have that offset, we have a monophasic and a biphasic one. And of course, this represents the window that we're looking at, the width of the sliding window. And what AdaBoost does is it will just try every single possible version of each of these kinds of wavelets, where you can vary the width and position of this phase, or this square wave. You can vary every possible combination of the width or separation of these by these two phases. So AdaBoost will try all of those. You take the dot product of this with your signal, and then you apply a threshold. And if it's depending on if it's above or below, you declare that exemplar to be either automatic or manual. And that's one weak classifier, right? And because there's so many possible configurations of this, you end up with many millions actually of weak classifier, because you can apply each one of these to the data from each sensor and every pairwise combination or difference, every pairwise difference between two sensors. This is what some of these might look like, right? So if you apply like this monophasic wavelet, then you get the area under here. And if you apply this biphasic one, you're ending up, basically what you're getting is a temporal difference, right? And the one I'm not showing is the offset. There's just the flat line, and that just gives you the bias, the mean in that time window. Here's an example feature. So let's say we're looking at the temporal difference, at the pairwise difference between those two sensors. We apply this, take the dot product of this wavelet against that within the window. And then you have a threshold on the output of that, which is a scalar value. So in general here, one weak classifier is feature plus threshold. So these are called decision stumps. So we're typically in boosting, you use decision trees. That's very common. We're not building a tree. We're just using one rule. And then, of course, this entire process is repeated separately at each position of the sliding window and for every possible combination, or every possible version of the three different kinds of our wavelet. And of course, trying this at all different positions, for example, this particular biphasic wavelet, trying it at all possible positions within the window, that just amounts to a convolution, roughly a convolution operation. And that if you're clever, you can code it right so that even though you have many millions of features to try, it can be quite fast to run. So if we look, these are in the data from the first three subjects from whom I got a large quantity of trials. And this is what the readiness potential looks like if you just take that time-locked average. And here it's a little more sophisticated than just recording from a particular sensor. What I've done is just built a spatial filter on the temporal difference between what's happening early and what's happening close to the movement. Here's what the spatial filter looks like. And if you project the data onto that filter, you get this readiness potential. And this is just to show, we get this highly replicable result. We get this nice readiness potential that stretches back arguably even almost up to 2 seconds prior to the onset of movement. So the question is, does this, let's say this point here, 2 seconds before the movement, is that reflect a decision to move? If that does reflect the decision, and this is certainly, we can resolve this quite well. If you go, say, just a second in advance, you're really far from the noise floor. So if this reflects a decision, then it should be predictive. And that's the essence of the question we're asking. If we do AdaBoost now in this sliding window, can we predict? We should be able to predict like a second in advance, a half a second in advance, and so on. At least that's what the readiness potential would suggest. But when we actually do this, and so here's an example so in white here, you have the sliding window. Let me turn off this laser pointer. So this white bit here is the sliding window. So what AdaBoost is seeing in this little, this is a video animation. What AdaBoost is actually seeing is just what's inside this white window here. And these are just two example features, an average feature that is negative for movement, and then in blue here, an average feature that's positive for movement. So as I animate this, just pay attention to the ROC curve here, and you can see how that evolves as the sliding window approaches time 0 here, which is when the movement actually happens. So here we go. So I'll play that again. So you can see it again. And what you might notice is that as the sliding window is sliding over the early part of the readiness potential, you don't seem to be getting any purchase until the sliding window actually gets to about 150 or so milliseconds before 0. Let's look at it again. There's really nothing happening until about now. And so if we look at the averages, we look at the results of doing this over, these were these first 3 subjects. You see that AdaBoost is not doing such a great job of telling them apart far before 0. But at the roughly the time of movement initiation, or maybe 100 or 150 milliseconds before, then it starts to be able to do so. And just at and after the onset of movement, it can do it extremely well. And these two, by the way, I believe the red line is the MEG data, the blue is the EEG data. So we've since run a number of more subjects. Here's 10 more subjects that we've run recently. This is just EEG data, but you can see once again, it's not really different from chance until you get about 150 milliseconds before movement onset, and right there and thereafter it goes up quite high. So it's not as if the machine learning algorithm isn't able to perform well on these data right after movement onset. It performs exceedingly well, upwards of 90% correct, and you can see even the individual traces here. And by the way, here in this case, we're comparing in red a growing window versus a sliding window, and performance is about the same. We expected that, but it was just a sanity check. So I mentioned before that there is this trick you could use if you only had data that was aligned to a movement, you didn't have any control data. You can, and this is what others have done in the past, you can say, well, I'll go way back in time and call this little window a baseline, and then I'll look at all these other epochs of the same width, but getting closer and closer to the onset of movement, and just keep comparing those back to this baseline. And asking, for example, with machine learning, can I tell them apart? And now, conceptually, now hopefully you agree that that's biased, and it's giving you an unnaturally optimistic view of how well you can classify, or it should. It's what we predicted, and when we did the analysis in this way, and here it's shown in red. So when we only looked at the movement epochs, and did the machine learning comparing each position of the window to a remote baseline, then we end up with, on average, this red curve. So it looks as if we can classify very, very well, even very early on. But we're arguing that that's a false impression, that that's cheating, so to speak. And the real difference, or the real ability to classify, is given by the blue line, which is the control method, which is where we compare actual movement plus slide transition epochs with just the slide transition epochs. So the manual versus the automatic trials. Again, this is with a more recent round of 10 subjects that we recorded using EEG. And here are the individual traces from those 10 subjects. So you can see it holds up pretty well for each one, and we're doing another round of 10. I think we're almost done, but I have little doubt that we'll have the same pattern of results for these other 10 subjects. So the evidence seem to support this late decision account more so than an early decision account, and we've been able to show that probably among the reasons that people thought this was an early decision was just because this kind of sample where you align to the onset of a movement is biased. And so moving forward, what we're trying to do is to look at this data in a very different way. And this is a technique that we've been developing, which is to, rather than, so that you can think of every point in time in this readiness potential where you just take the average time locked to movement. Every point in time at the readiness potential reflects the distribution of the probability of the signal now given a movement at a specific time in the future, right? And what we want to do is look at that the other way around. We want to look at the probability of a movement at various times in the future, given the state of the signal now or in the recent past. Something more like building a weather forecast in a sense, or a forecasted trajectory, say of a hurricane, where you out X number of days in the future, you're predicting where it's going to be or what its state is going to be. And the nature of that distribution can actually tell you something about the degree to which the process is stochastic or deterministic. So that's where we're at right now. I just wanted to share this work with you, and hopefully start a discussion on these kind of general topics. I do want to point out that I started this work back when I was working at Neurospin, and I had funding from the European Research Commission and a ERC starting grant, and I obviously have since continued the work now here at Chapman at the Brain Institute. So thanks for your attention, and I open it up to discussion.